So structure of nucleic acids, right? Sounds exciting. The first figure I have to show you, okay, is central dogma. Now, central dogma says we go from DNA to RNA to protein, and we go through transcription and translation, okay? And as you can see, in the central dogma, 2 of the 3 stages of the biomacromolecule are nucleic acids, okay? So DNA is a nucleic acid, RNA is also a nucleic acid. So nucleic acid is quite important, even though it's not exactly super exciting. So DNA, obviously ATCG, RNA, AUCG. Okay, now, chemically speaking, your nucleic acids are made out of 3 things. You have a pentose sugar. We'll talk more about sugar chemistry when we get to carbohydrates, but for now, just understand what I'm trying to say. It's a pentose, meaning it's a 5-carbon sugar. Okay? Pent, like a, pentagon or penta, whatever. Okay? Pentose. For RNA, it's ribose. For DNA, it's deoxyribose. Okay? You also have a phosphate. Okay? Somewhere. Okay? Next to the 5 prime, of course, 5 prime carbon. Okay, we'll get there. You have a phosphate. And you have an organic nitrogenous base. Okay, this is the DNA or RNA base that we usually talk about, the ATCG. That's the nitrogenous base. Okay, so let's real quick take a look. So for RNA, which is shown on the left, and DNA is shown on the right, what you see here is a couple of things. In blue, that is your sugar. Phosphate is the thing right here. That's the phosphate. It's always attached to the 5 prime carbon regardless of DNA or RNA. It's always attached, the phosphate here, okay, this bond right here, this guy, is always attached to 3 prime OH, 3 prime hydroxyl. That's why we say DNA is always 5 prime to 3 prime. Okay, now, okay? At this moment, you probably will not understand why this guy is carbon number 1 and why this guy is carbon number 5. The carbon that is not in the ring is carbon number 5. So that makes this guy carbon number 1. Why? Because you can't go 5 to 1, right? So it's always 5, 4, 3, 2, 1. And this guy's an O. RNA, it's a ribose, okay? Ribose will be a sugar, and sugars will always have OH on every carbon except 1 carbon. For ribose, the 2 prime carbon has an OH group, so that's the normal sugar. Okay, so that's RNA. Okay, this guy, the thing in the blue, is the ribose. Okay, that guy is the ribose. When you get to DNA, it's called deoxy, okay, removing oxygen. So your 2 prime OH is gone, okay? It's like reduced, right? It turned into a simple hydrogen, okay? It's gone. That's why it's called deoxyribose. RNA, DNA, okay? And then we talked about how because RNA has that extra OH and usually it's single-stranded, it forms secondary structures, remember that? So nitrogenous bases are divided into 2 types. We have the purines and the pyrimidines. So purine derivatives, why they're called purines? Because purine is this guy right here. Purine derivatives are adenine, guanine. Okay, yeah, you don't have to memorize the structures, okay? But you should know that purines have 2 rings. Okay, please at least memorize that. Pyrimidine, as you'll see, has only 1 ring. Okay, so please just remember purines have 2 rings. Okay, 1 5-member ring, 1 6-member ring. So adenine, you have essentially amino group on the top, and that's pretty much it. You have amino group on the top, and then you have these 2 nitrogens right here. Guanine, on the other hand, you have a keto group on the top, and then you have an amino group at carbon 2. There's little numbers written in the ring that tells you which position that we are talking about within the purine ring. Okay? But for adenine, essentially you only have an amino group on carbon 6 or position 6, and then for guanine you have a keto group at position 6, amino group at position 2. Okay, so obviously the shape is different. Pyrimidines, you have 3 pyrimidines. Okay, 2 are used for DNA. Actually, no. 1 is used exclusively for RNA. 1 is used for DNA. Cytosine is used for both DNA and RNA. Okay, so you got cytosine, thymine, uracil. They're called pyrimidine because they're sort of derived from this molecule called pyrimidine. It's one ring. Okay, so cytosine, if you look at it, you got amino group, you got a keto group right here. Thymine, you still have the keto at position 2, but instead of the amino on position 4, now you have a keto group right there. Okay, so you got 2 keto groups. Thymine has 1 extra, what is this guy? Methyl group, that's right. This is a methyl group. Okay, that's a methyl group. So thymine and uracil looks very similar because they have the keto, except uracil does not have the methyl. So it is not surprising to you guys that back in general biology you learn ATCG for DNA, AUCG for RNA. So T is replaced by U in RNA. Right? Because T and U, they look pretty much the same thing. Now, methyl groups, okay, methyl groups essentially only changes the shape of the molecule. Okay? It does not provide any bonding features. Remember, we talked about electron bondings, right? Polar, that kind of stuff, right? It does not form any hydrogen bonds. It does not have any electrostatic interactions, or at least significant ones, okay? Because it's hydrophobic. Doesn't do much, the methyl group. Right? So you have U and T, pretty much the same thing. Ribose ring attaches to your position 9 right there. For purines, for pyrimidine, ribose ring attaches to position 1. Okay, so what you notice is your ribose ring always attaches to a nitrogen. Okay? It's always attached to a nitrogen. Pyrimidines, you don't have to worry too much because you have 2 nitrogens, right? And obviously, it's not going to be the nitrogen between the 2 groups. It's going to be the other nitrogen. Okay, so just always remember that, right? Because it does not make sense if the nitrogen, if the ring is attached to here. No, so it's always attached to here. Okay. For purines, please always remember the big ring faces outside. Okay, that's simple, right? So the big ring faces outside. Okay, faces outside. Okay, so the big ring is used to do pairing. That should be pretty simple. So we got nucleosides versus nucleotides, okay? When we say we're talking about nucleotides, usually, nucleotides have phosphate. Nucleosides do not. Let's get the names straight, okay? So I think the names are important. You don't have to memorize the structures for me, but please at least know the names. Bases, we have adenine, guanine, cytosine, uracil, or thymine. This is RNA, right? So uracil. Nucleoside, meaning after you attach the base to a ribose, you attach it to a ribose, then you have a nucleoside. Adenine then becomes adenosine. Guanine becomes guanosine. And cytosine becomes cytidine. And you got uridine. That's nucleosides. After you activate the nucleosides and you make it become nucleotides, then the name is actually quite simple. It's just adenosine 5' monophosphate, okay? Because this guy here is adenosine. Adenosine this carbon here is 5' carbon. So adenosine, 5', and then this is a monophosphate because you have only one phosphate, monophosphate. Okay? Alright, so adenosine, 5' monophosphate. Okay? Guanosine, 5' monophosphate. Cytidine, 5' monophosphate. Uridine, 5' monophosphate. Okay? Now, we usually are lazy, so we say AMP, GMP, CMP, UMP. Okay? MP usually just indicate that it's only one phosphate. Okay? So if you have DP, it means diphosphate, two phosphates. TP meaning triphosphates, that means you have two more. So that's RNA. This is DNA. DNA, everything above this line were the same as the previous slide, except here. Except the 2' OH is gone. Okay? We got same thing, adenine, guanine, cytosine, thymine. Now this guy's thymine instead of uracil. Okay? Your nucleosides, now instead of adenosine, it's deoxyadenosine. Oh, great. Okay, so it's deoxyadenosine, deoxyguanosine, deoxycytidine, deoxythymine, thymidine. And then for the nucleotides, you have deoxyadenosine 5' monophosphate, DAMP, DGMP, DCMP, DTMP. Pretty straightforward. Turns out, okay, you got some tautomerization of nucleotide bases and conjugation of nucleotides. Okay, what does that mean? All right, so let's check this out. Adenine, here this guy is an amino group, but next to this carbon, you got a double bond to nitrogen. Okay, so what you can do is you can do like a little movement in hydrogen such that the electrons, lone pairs here, take this electron, the electrons here, come down here, okay, and this guy actually goes up. Okay, so the electrons are balanced and you generate this different structure for adenine. If adenine can do this, cytosine can do this, okay? Because both of them have this amino group next to a double bond N. Guanine, on the other hand, can do a keto-enol tautomerization. Right? Essentially, similar idea. You have a keto, you can become an enol if by you sort of move the electrons from this guy to this guy go up, and this guy grabs the proton, and then you have an enol. Typically speaking, most of the times, even though your bases have two forms, but most of the time you'll only see amino and keto. You don't see the enol, okay, because enols are generally less stable than keto. Okay? So you will generally always see the amino form for example, this guy, and the keto form of, for example, guanine, right? You always see the keto. Always see the keto, right? Instead of the enol. It's just to show you that DNA bases have two different forms, but the amino and the keto dominates because they're more stable. Because these nucleotide bases have conjugated double bonds, conjugated double bond means like this, right? Like that, alternating double bonds, meaning it's conjugated, okay? And you know that as long as you have conjugation, you will absorb light. Okay, so because DNA bases have conjugated double bonds, so then they can absorb light. This turns out to be a super good thing for us because we can use absorbance of UV light to quantify DNA. This property of nucleic acids are often used to quantify the concentration of DNA or RNA in a laboratory. Okay? Because of the conjugation, you can absorb light. Because you can absorb light, you can quantify. Because it absorbs light, okay, because it has these double bonds and double bond pi electrons, they are easier to be excited, kind of, maybe. Okay? So, turns out You know from commercials about how like excessive UV light will cause damage to your skin. That's why you have to wear sunblock, you know, that kind of stuff, okay? This is the reason. Turns out UV light will cause formation of a new carbon-carbon bond between adjacent pyrimidines at C-C double bond. Okay, what does that mean? Okay, so this is the bond that I'm talking about, C-C double bond. So let's take a look at this particular picture. Okay, you have thymine. Okay, because you have 1 keto, 2 keto. That's thymine. So you have two adjacent thymines next to your DNA. Okay, so you have like, for example, A, T, T, A, G, G, blah, 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 T, T. If you have excessive amount of UV and if the UV energy is high enough, you can actually trigger a bond breaking between the CC here, such that you will form a new bond between this carbon and a neighboring carbon. Initially, you have a bond. Shine UV, this bond is gone. Okay, gone. And then you formed a new bond with this guy. Okay, and you can do it a couple different ways. Right? You can do it, link them together like that, or you can link them together like that. Okay, so it really depends on what you have right there. Usually in mammals, especially higher organisms, we have mechanisms in which we can repair this problem, because obviously the second you walk out in the sun, you have a lot of UV ray hitting on you, and you got DNA all over your body. So as a result, of course, your DNA is going to get mutated. So then we have this process called the nucleotide excision repair that is used to identify and remove UV damage. Okay, why is that? After you form the dimer, your DNA will actually have a little hump like this, okay, because you physically link two thymine units together. So actually it will cause the DNA to have a little kink, okay? So because of that, there's a cellular process that can run through the DNA. As soon as they realize there's a problem right there, they will cut off this entire region, they'll cut it off, and then repolymerize it, okay? Fix the DNA sequence right there. Okay, so your body does that. That's how we're able to withstand UV damage from the sun. This usually only happens to pyrimidines, so you can get TT dimers, CC dimers. Let's talk a little bit about how we actually form polynucleotides. Okay, so we talked about nucleotides, right? So nucleotides has one phosphate. Okay, turns out, okay, the subunit for making DNA is not the monophosphates, is actually the triphosphate. Okay, so the actual addition subunit for adding DNA bases onto a growing chain is using nucleoside triphosphate. Okay, for example, DATP, DTTP, DCTP, DGTP. Has to be the TP, triphosphate. Okay, why is that? Okay, why is that? Let's look only at this line for a minute. The energy is positive 25. If you were to only use a nucleoside monophosphate to add up a base onto the growing chain, energy is positive 25. Yeah, it doesn't happen. Okay, it just does not happen. So in order to make this reaction, the overall reaction of adding a DNA N to become DNA N plus 1, you have to give it a higher energy building block, which is the DATP, DTTP, or in general we call DNTP. N can stand for whatever. Okay? So direct addition of nucleotides to growing chain of DNA or RNA is thermodynamically unfavorable. Okay, so instead of using nucleotides to add to a growing chain, we actually use nucleotide triphosphate. That you have to know, because if you want to directly add nucleotide onto it, energy is not high enough for you to do this chemistry. Okay, so you have to use nucleotide triphosphate. So for example, ATP or deoxy ATP are used, and in this process you liberate pyrophosphate instead of you don't release anything. Okay, so you actually, the release of the pyrophosphate is a favorable thing that happens. Nucleic acids are metastable compounds that are thermodynamically favored to break down, okay, but do so only very slowly unless the reaction is catalyzed either by acidic or basic treatment or by an enzyme catalyzed reaction. And the enzyme is called nuclease. Okay, so DNA, it's actually, if you want to break DNA, it's a favorable chemical reaction, right? Because you liberate a base, right? And then you have a stabilization of the charge, right? Because otherwise your DNA has a lot of negatives, right? So if you break it away, you have a reduce in the repulsion, kind of. That's number one. And the fact that you generate extra molecules. So this breaking of DNA is favorable, but the reaction speed is very slow. If it's very fast, you and I will not be alive, right? Because like your DNA will be spontaneously degrading, okay? However, if you throw acid or base into the mixture, then it degrades very fast because you have a change in pH. Let's take a look at the pictures. Right? Pictures are more valuable in this sense. Right? So this is the formation of a polynucleotide by a hypothetical dehydration reaction. So this is a hypothetical. Okay? This is what we usually teach to little kids, but you guys are second year or third year college students, so this is not true. Right? This is just hypothetical. Hypothetical reaction goes like this. We have a growing chain polynucleotide with n number of residues. I add the n+1 residue. Okay, so this is one monomer of DNA. This guy is just a monomer. Okay, and I make it into n+1 residues. Okay, so I polymerized it. Okay, this is not favorable. Okay, as you can see, indicated by this tiny arrow for dehydration. So this polymer plus the nucleotide monophosphate monomer to give you this, not favorable. Okay? The reverse is favorable because this plus that to this, energy is positive 25. So the reverse energy is negative 25. So in reality, we want to go from DNA polymer to DNA monomer. We want to break down DNA. At least that's the chemical equilibrium. This is only a hypothetical, right? So it doesn't work this way. The polymerization of DNA does not work this way. You have to use the triphosphate as your actual monomer that adds on to your nucleotide. So this is how the polynucleotides are actually formed. Each monomer is presented as an NTP to be added to the chain. Okay, again, so this is the growing chain. And then this is the monomer. It's the 1, 2, 3 triphosphate. And when you do this reaction, you cut off this bond, this phospho and hydride bond. Okay, you cut it off, okay, because here you have a lot of energy stored in here, right? You have a lot of energy stored in the triphosphate. You cut it off, you liberate pyrophosphate, and then the phosphate is now going to be attached to the 3' end of the previous space. And because the hydrolysis of triphosphate gives you a large amount of energy, that's how you're able to push this reaction forward. The numbering system, it's always DNA, you always add from 5' to 3'. This is the old 5', right? The old 5'. The old 3', you add a molecule, you add this guy on here. Now you have a new 3' hydroxyl that's free that can take another 5' phosphate. Okay, so DNA polymerization is always 5' to 3'. Okay, because you cannot possibly do 3' to 5' because the 5' is just a single phosphate. Energy is just not enough. Okay, so 5' DNA to 3', 5' here is a phosphate, 3' here is OH. If you want to add from 3' to 5', here is just one phosphate, it's not triphosphate, okay? So energy is not high enough to do the addition. So monomer is always added from 5' to 3'. You always add to this guy. So because of that, polynucleotides has a sense or directionality. Okay, so DNA has a direction. The phosphodiester linkage between the monomer unit is between the carbon of one monomer and the carbon of the next. Okay, thus the two ends of a linear polynucleotide are distinguishable. Okay, so for example, we have CTGA. This is very different from AGTC, okay, because this guy is 5', this guy 3', 5', 3'. Okay, so this guy has a phosphate. This guy has an OH. This guy has an OH. The phosphate, okay, the A phosphate is attached to the G. Okay, now C has the phosphate, okay, instead of the OH. So DNA has a direction. So one end normally carries an unreacted phosphate, 5'N. The other end carries an unreacted hydroxyl group, 3' OH, 5' phosphate. Okay? So a polynucleotide chain has individuality determined by the sequence of its bases. That is the nucleotide sequence. So all this is saying is A, T, C, G, G, C, blah, blah, blah. It's distinguishable because the sequence, for example, ATCG is different from ATGG. Okay? It's just different. It's different molecule. And the sequence is called the primary structure of nucleic acid. For biomacromolecules, primary structure for DNA and proteins is always just the sequence. Okay? The order at which things come. Secondary structures we'll see has to do with intramolecular bonding, hydrogen bonding, and stuff like that. But primary structure is just the sequence. Okay, now, some people actually prefer to have a notation that looks like this. For example, A, C, G, T, T, and then they show how you have a phosphate. 3' of A binds to 5' of C through a phosphate. That's just annoying. Everybody's purpose, we always just write A, C, G, T, T, and then we know exactly what it means. Okay? If I want you to draw the structure of A, C, G, T, T, you should be able to do it, minus the bases. Okay? I don't care if you memorize the bases or not, but you should be able to reproduce the ribose ring, the phosphodiester linkage. Okay? You should be able to reproduce that. And you should know the difference between RNA and DNA. Okay? So if I want you to draw a structure of A, C, G, T, T, you should be able to do that. Essentially, this notion of this whole DNA sequence shows that the sequence of nucleotides by their letter abbreviation. Okay, so we can use their base abbreviation to show what it is. Okay, all phosphodiester bonds are between the hydroxyl and the phosphates. That's true. This particular molecule has a phosphate group as 5' N and unreacted hydroxyl group as 3' N. So 5' N has a phosphate, 3' N has an OH. And it also tells us it's DNA sequence, not RNA, because it has T and not U. If I give you something that looks like that, you should be able to tell me that it's a DNA and not an RNA. That should be very simple. The primary structure of nucleic acids is nearly always written from 5' to 3'. In general, we just always write it this way. Okay? We don't use this. We don't use this. We always use simple ATCG. And the one on the left is just 5' N. The one on the right is the 3' end. Okay? That should be pretty straightforward. All right. So the main importance of a DNA sequence is that the genetic information is stored in the primary structure of DNA. Hold on. A gene, right? So every time we talk about a gene, Gene is nothing more than a particular DNA sequence, which encodes an information in a 4-letter language, which each letter is one of the bases. Okay, so that's it. Right? So genes are just a sequence. Right? So this is something that molecular biology will actually talk more about. Now, for biochemistry purpose, I want you to be comfortable with the chemical structures of the DNA linkage. The bases are okay, because I don't think most people will memorize the DNA bases, but you should be very comfortable with the structure linkage of the DNA. So from this part of the lecture, you have to know a couple of things. Difference between DNA and RNA, you should know. DNA, RNA always go 5' to 3', and most importantly, the actual building block for DNA and RNA is not the AMP, DAMP, it's the triphosphates. Okay, that is a very important message. Okay, so why is that important? Because remember we talked about ATP. ATP tells you whether or not your cell has energy. Okay, so your cell will not be making DNA if it does not have energy. Okay, so it all makes sense. Cool?